All right, well then you are here, everyone, to do some amazing hands-on science. And thank you again for joining us. I'm going to toss it over to Sam Grant, who can go ahead and introduce himself and tell us about what we're going to be up to tonight.
Hi, folks. My name is Sam Grant. I'm a professor of chemical and biomedical engineering at the FAMU FSU College of Engineering. I'm also the director of the MRI user program at the National High Magnetic Field Lab. So welcome to eWeek and welcome to the MagLab and College of Engineering's virtual open house this year. Obviously, because of pandemic concerns and the like, we're not able to do demonstrations for you in person as we usually love to do. But hopefully you'll get a flavor of that and be able to do some of these types of demos, particularly the one that I'm doing today, at home along with me. So the demo that I'm going to do today takes roughly about 30 minutes, can vary a little bit in time, but we'll give you effectively all the details and all the information that you need in order to do this at home. And you can wait and hopefully be able to see some of the DNA that lives inside the cells that make up your food. So we're going to first jump right in with a couple of the items that we need in order to do these types of experiments. And I'm going to start with the first one because this is something that needs to be pretty cold. So if you don't already have this in your freezer, I recommend that you take isopropyl alcohol, rubbing alcohol, the kind of things that we use when we're cleaning a wound or something of the like, and go ahead and put that in your freezer. You need this to be fairly ice cold in order to help us with the DNA extraction, but this comes at the end. So take a second and go ahead and put that in the freezer. We're also going to use a couple of other items here. Number one, we're going to use regular table salt. So regular white table salt will work for us. We don't need very much. We only need effectively something on the order of about 2 grams. 2 grams is roughly about half a teaspoon or so. And we can be a little bit inaccurate in the measurements, but you don't need very much of that. We're going to need some dishwashing soap as well. Anything will pretty much work. Something that doesn't have color is nice, but even something that kind of is green, as a lot of dishwashing detergents are, will be fine too. We're going to use this as a means to actually break up the cells in the fruit that we're going to be looking at, so that we can get at the DNA. In addition, we need some coffee filters. Doesn't make any difference what kind of coffee filter you have. It can be kind of the flat coffee filters or the cone coffee filters. You can use either. These are going to help us to purify our solutions and do a little bit of simple separation so that we can look at our materials a little bit later. In addition, we need some bags that are zipper sealable. Any kind of bag will do. These are sandwich bags, pretty simple, straightforward things. 1 or 2 of these will help with your experiments. We need water, just regular tap water, although it's best if it's a hot water in a confined amount of volume. We only need about a half a cup or roughly 100 milliliters of water. So if you have that, that's great. If you have additional hot water, it helps to speed up the process a little bit, and I'll show you how. Measuring cups, and then also just some simple kinds of glasses. Can be any type of glass, really. Here's basically a jam or jelly jar. Here's a baby food container. Here is a champagne glass. Anything that's kind of tall will work a little bit better so that we can look at the extraction of the DNA that's coming off of our solution. And then finally, minus a couple other things like paper clips, which are pretty easy, mixing, stirring rods. The stars of the show today are going to be fruit. Fruit is basically just an easy, accessible way for us to look at DNA because we need something that has cells. We need something that we can grind up pretty easily so that we can look at that DNA and extract it. So strawberries work really well. If you're using a strawberry, just make sure that you pull off the leafy green part and use that. Bananas also work really well with regards to this. You don't have to use a whole banana. You can use a half banana, so you can eat the other half if you're hungry. I know I am, although I'm not going to eat in front of you. But we'll use about a half of this banana to get started. And then what I'm going to do is I'm going to put the banana into a bag. And then I'm going to try to squeeze all of the air out of that bag. Okay. All right, so I have something that's kind of air-free and looks something like this. Okay, and now the fun part. I get to mash up the banana. And you want to beat it up pretty good because we want to basically destroy all those cell walls. We want to get into get past the membranes eventually and get into the nuclei of the cells so we can get the DNA out of those nuclei. So we want this to be, fairly well mushed up, and you're going to want to do this for a couple minutes in order to get it nice and ready for our extraction technique. Now, of course, don't beat it up so much that the bag opens and we make a big mess or anything like that, but get it so it's nice and mushy. And effectively, you want something eventually that's going to look like the consistency of pudding. Okay. All right. So we're getting there, and we're going to keep mushing this up for a little bit. While that is being mushed up, if you have a parent or a brother or sister or a friend who can help with this, that would be nice. We're going to make up a saltwater solution. The salt water is going to help us get into the cells. It's going to help us to get the DNA out. So we're going to use just one of our jars. We're going to put in about a half teaspoon of salt into this. Again, doesn't have to be exactly accurate. We have some flexibility there, but half teaspoons would guess. And then we're going to put in some warm water to make the salt water a little bit easier to dissolve and to get into our cells. So again, about 100 milliliters of water will work. And then we will mix that up. The water I have here is about 60 degrees centigrade. So it's not boiling water, but it is definitely warm. And after I warm this up, we're going to pour it into our little fruit bag so that we can start to mix up that salt solution, which is going to help to prepare our DNA for extraction. Okay, so we're going to take our salt solution and we're going to very carefully pour it into the bag. Best to do this over the sink or to do it over a bowl. If you have access to one, that's usually going to be the easiest way to do it. And then we're going to close the bag. Again, try and get most of the air out. And then we're going to mix it up again. And we're going to beat it up some more so that those cells are what's called lysed, so that they're separated and they're nice and open so that we can get into the nuclei of those cells. Now, the salt water helps to maintain the DNA, helps us to get at it a little bit more accurately, but it's not going to get us all the way home. We need some type of chemical in order to help get into the cell membranes and get into the nucleus and break up some of that cell structure that's in the cells of this banana. So what we're using is effectively the dishwashing soap so that we can do that. Dishwashing soap is what we refer to as a surfactant. Surfactants really get in and disrupt lipids in particular. And so the cell membranes, the cell walls in this case, as well as the nuclear membranes, are made up of lipids. So by adding a little bit of our dishwashing soap to this mixture, we're going to be able to mix it, get in and break up those lipids and hopefully get the DNA out. So in this case, you want to have basically about 5 grams of dishwashing soap that you put into the mix. 5 grams is equivalent to about 1 teaspoon of dishwashing soap. So that's 1 teaspoon, we'll put that in, okay? Now, what happens when you add dishwashing soap to water? It starts to get bubbly, right? And that surfactant goes to work and it starts to basically break up lipids, which is good if you're washing dishes. It also is going to start to interrupt the surface tension that is on your dirty dishes and therefore allow you to clean the dirt off your dishes. But it's also going to make bubbles and it's going to make foam. So we want to mix this solution real carefully without creating a lot of bubbles. Bubbles are going to cause us a little bit of a headache later on, so we want to try to avoid that as much as possible. Okay, so we're going to mix that real carefully. Ultimately, that surfactant is again working its way into the cell membranes and the cell wall in this case because it's a plant, and into the nuclear membrane, and that's going to free up those lipids and allow us to get at the DNA. So now we have hopefully in this gooey mix we have DNA that is dissolved in the water and is floating in our mixture. So the combination of the salt water and the dishwashing fluid effectively makes our DNA extraction fluid, our chemistry, for this type of demonstration. Okay. If you want, you can also take your bag and you can immerse it for a little bit of time in hot water, which I'll do here. That kind of speeds up the process a little bit. So I'll just let it float there for a second. And then while that's floating, we can talk a little bit about DNA and why I, as a researcher, am concerned about DNA and study it to a certain degree. It's not my main area of research, but I'm a biomedical engineer. I also work at the MagLab, utilizing the high fields there in order to perform very high field magnetic resonance imaging, or MRI. We look at a lot of models of neurodegeneration. So we look at things like acute degenerative diseases like stroke, as well as more chronic diseases like Alzheimer's disease. And in many cases, what we're doing with those different diseases, whether we're just analyzing the disease or trying to develop new treatments for the disease, is we have to be very concerned about the genetic material of the models that we use or the cells that we're using in those models. So, for example, for stroke, we use stem cells in order to try to recover from that stroke more accurately, more effectively, more functionally. And one of the things that we can do is we can condition those cells in different ways in order to force their DNA to actually express different types of proteins to carry out different types of functions. And so we can do non-viral, non-damaging techniques that don't actually directly change the DNA of the cells, but allow them to encode different types of instructions to produce certain proteins, to produce certain types of helpful chemicals that have an anti-inflammatory, a pro-regenerative type of response, or even something that will help to grow more blood vessels in that stroke area. We also deal with models like Alzheimer's disease. And Alzheimer's disease, when we look at an animal model, is a human transgene that has been transplanted into the genome of the mouse. So effectively, we've taken genetic material from an Alzheimer's patient who has a genetic alteration, and we've introduced that into a mouse model so that we can study the progression of those diseases. So all of these types of techniques we can analyze using MRI in order to track the progression of the disease and how well our treatment is actually affecting a change that hopefully recovers functionality. Okay, so now we waited a little bit. Our solution is nice and warm. I've made a few more bubbles than I probably would like to, but that's okay. Next thing we're going to do is take our solution and we're going to filter it. So we're going to use the coffee filters in order to do that. And if you have a tall glass, that works. Even if a short glass, that's fine. We're going to put the filter into that glass, and then we're going to very carefully pour a mixture into the filter. And we're going to let it drip through the filter into the empty clean glass. Now, this is a little bit tricky again, so be careful. Make sure that you if you're a younger MagLab and College of Engineering open house visitor, that you have a parent that helps. You obviously can use a basin to help catch up any of the spillover if there is any. And then we're going to let that drip through the filter. And what's happening is now we're basically removing the solids from our mixture, and we're just getting the dissolved materials that are coming through as fluid. Okay, you need to be a little careful at this stage so you don't rip the filter. But if the coffee filter does rip, just take the material that fell into the glass and just filter it again through another clean new coffee filter, and that'll allow you to get all the material out. So this does take a little bit of time, and it takes a little bit of effort for that to get through. So you're going to have to be a little bit patient as you let this mix through. If you can push it through a little bit with just a spoon or something like that. But again, you have to be fairly careful not to rip the filter. Okay, so we'll let that work for a little bit. Pour a little bit more in of the fluid, not so that it goes over the top, and then we will seal the bag up. And as you can see, we're starting to drip through and collect liquid at the bottom. Okay. All right. So now what I've done is, before we started is I went ahead, just in case, and to demonstrate this a little bit more clearly, I went ahead and took some strawberries. Okay, and those strawberries now I've filtered through a coffee filter, and we have a nice bit of volume that is has come up there. Let's see if we can get a little bit more fluid out of that from our strawberry bag. So I'll pour a little bit more on. All right. And we'll put that in the hot water so we can get some more out of that in a little bit. And then we'll press down on this filter, again, being careful not to rip the filter, so that we can get some of that nice dissolved liquids through the material. Okay. All right. Squeeze it a little bit, get a little bit more out, and then we'll put that over there. Okay. So now we have our purified fluid. We have the banana still working, but it's starting to come out nice. It's mostly a clear fluid with a little bit of yellow color, cloudiness. If you're using strawberries, obviously that, because the color of strawberries is a little bit more pink, but that's all good, that's all okay. Now we're at the extraction phase, and this is where you need to be able to go back and get that cold rubbing alcohol, that isopropyl alcohol from the freezer. I'm going to run to my freezer and get some out, and then we can start that process. So it's a good time to let me ask if there are any questions. I'm happy to answer those.
I don't see any questions in the chat right now, Sam, but while everyone's getting their alcohol, I hope you're following along with this amazing fruit extraction process. This is so much fun, Sam.
Okay, thanks. All right, so next step is we're going to use our cold alcohol. And what you can see is I've gone to a document camera. You can see my fingers, you can see the glass here. And I'm going to use alcohol and pour it over top, okay? So when you are pouring this, in order to extract the DNA, you want to make sure that, and let me switch back to the other view so you can see this a little bit more easily. You don't want to mix the alcohol. You want to pour it so that you're more or less running right down the side of the glass, okay? So I'm going to very carefully take my alcohol and I'm going to pour it down the side of the glass so I don't mix. Okay, and you only need about 100 mLs of alcohol in order to do this, so roughly the same volume as you have with the solution itself. And what you can see, if you look very closely, is that you will have the alcohol floating on top of the water solution. So let's see if we can see that. So if you look real closely here, you can see that the alcohol is floating right on top of the water. Okay, I'm going to put a little bit more alcohol in. And hopefully you can see that as I do it here. All right, again, I don't want this to mix. I just want to float the alcohol on top of the water mixture. Okay, so we've got a nice volume there. Okay, of alcohol on top of the water. Okay, and now we wait. And what we're waiting for is basically the DNA to come out of solution. What's going to happen is basically, because we've used cold isopropyl alcohol, that alcohol is serving to effectively cause a point of nucleation, which means the DNA, which is dissolved in the water, does not dissolve in the alcohol. So at the interface of the water to the alcohol, we're going to start to see white strands of DNA that are going to come out. And those white strands are going to coalesce into something that we can see pretty clearly. Now, you don't want to mix it. You just want to keep the interface there pretty clean and pretty uniform so that the alcohol is floating on top of that mixture. But you can already start to see in this mix some of those white strands come out. I'll see if I can turn it on the side again and kind of focus in here a little bit. And you can start to see white cloudy areas that are coming out at the interface between the water and the oil. Okay, those white strands are basically the DNA now no longer dissolved, but coming out into the alcohol. And that's how we can basically extract the DNA. Okay, and we can use a lot of different things. If you have paper clips that are handy, you can take a paper clip and you can dip it into your solution. We'll see how far along we are here. And if you're real careful, and you have enough of it, you can pull out some of those white strands. Okay? So you can give that a try. It does take a little while for this to happen. So it's been a couple minutes now in our solution, and I can start to see the white strands actually in the alcohol. And as we wait longer and longer, there'll be more DNA that comes out of the solution. And if there's enough DNA, you can actually pick it up with your paperclip and be able to see it real clearly. Okay? So that cloudiness is going to get more and more. Those white fibrous materials there are the DNA.
Sam, can you focus that camera in for us? It got a little blurry there for a little bit.
Pardon me?
Can you focus the document camera? We're having a hard time seeing that.
Absolutely. So you can see a little bit from this that it's no longer clear at the interface, and we're starting to see more and more of the DNA coming out at the interface of the alcohol and the water. So that's the strawberry's DNA. That's the genetic information that encodes for the flavor of the strawberry, the color of the strawberry, the seeds that grow on the outside of the strawberry. All that type of information is encoded in the DNA. We do this in human cells when we're analyzing what kind of effect our culture techniques have had on our stem cells that we use to treat neurodegenerative diseases. And so we'll do extractions and then analyze what types of genes are being expressed, what kind of genetic material is there, and how that's being altered by some of our conditions so that we can understand really the mechanisms that are at play behind our types of techniques. So that's the strawberry solution. I'm going to go ahead and set this aside for a second. The banana solution also looks like it's got enough fluid here. So I'm going to go back and bring that over to the document cam. And we can see how that one looks. Of course, as I did this, I ripped the filter, but that's okay. I'll just use another filter. Okay. And so I've got this filtered through another filter. Okay, we've got some nice fluid that's there. Give it a little bit of a squeeze just to extract a little bit more. Okay. And then again, I'll pour the alcohol right on top of that, keeping the glass at a 45-degree angle or so, and just floating the alcohol on top of the, in this case, the banana DNA mixture.
Sam, we've got our first question coming in. They're asking, why are you able to see the DNA this way?
Yeah, it's a very good question. So what we have is with the addition of the salt water, we've protected the DNA so that it's able to come out of the solution into a comfortable environment. With the dishwashing soap, because it's a surfactant, we've broken up the cell components that are made up of lipids so that we can get at that DNA. And then when we put the alcohol on top, unlike the water, the alcohol, it does not allow for the DNA to dissolve. So as that, it has no choice but basically to come out of solution because of its presence in the alcohol. And as it comes out of solution, it condenses, aggregates effectively. And so it becomes those white strands of DNA. Normally when we're in solution or when we're in cells, unless there's a cell process going on, the DNA is actually very long and very dissolved. When we need to that genetic information, or in this case where we've artificially kind of forced it to come out of solution, then it becomes very coiled and very tight, and then aggregates in the conditions that we provide. So even in a short amount of time in this champagne flute, I can start to see some of the DNA coming out of the banana mixture, and we start to see these little white strands that are coming up. So it's a little hard to see in the background, but let's go back to the strawberry, and you can see even more cloudiness. Oh, the strawberries come out very nice, because now you can see a nice layer of strands that are coming out. Let me see if I can focus that a little bit better.
Yeah, I just saw that white layer. That looks really good.
Yeah. And so when that gets, if you let that go for a little bit longer, then you can use your paper clip or your toothpick or whatever you have handy and actually extract it. And you can bring that white gunk out, and that's the DNA of the straw. You can actually do this with cheek cells. It's not very COVID friendly right now, but you could scrape the inside of your cheek. You could look at the extracted DNA using the same technique, using human cells or other types of cells. And that's effectively what we do when we do some of our analysis for figuring out exactly how cells are affecting changes in our different models. So that's the demonstration. Any questions?
That was fantastic, Sam. I think that it's a great reminder, right? That I heard you saying at the end, we could do this with our cells, maybe in the safety of our bubbles, right?
Right.
With samples from your own mouth, and you would do everything else the exact same way as you did with the fruit.
Yep, that's right. And you can use different types of food as well. You can look at the cells that come off of, let's see, onions. You can look at any different types of fruits. Usually you want to use things that are fairly easy to mash up so that you can get at those cells and start to break up the cell membranes fairly easily. Things that are a little bit tougher or have a really hard cell wall are a little bit, generally a little bit harder to extract. But things like seed pods from green beans or other types of seeds work fairly well. And there's a lot of genetic material that's in those types of food products. You can take a look and see how what different types of foods extract more and more types of materials. And if you think about it, for an average human, we have trillions of cells in our body. And those trillions of cells have DNA that encode what those cells do, whether they're brain cells, whether they're muscle cells, bone cells, those types of things. But the total mass of the DNA that's in the body is only about 250 grams. So it's very little compared to the entire mass of the body, but very important because it regulates how proteins are made, it regulates how cells are developed, regulates how different types of processes are actually carried out in the human body.
Yeah, I just put it in the chat, Sam, but all this talk about fruits and veggies right in time for dinner does make me think of a new game we actually just launched. And it relates to a magnet system that you use, using some of those wonderful images from one of our coworkers at the MagLab to take MRI, high resolution MRI images and try to guess which fruit or vegetable it is based on the high resolution MRI. It's harder than you think, right, Sam?
Yep, absolutely. It's MRI is nice because you get a chance to take a picture of the insides of animals, humans, and in this case, fruit. And you don't really think about what your fruit looks like from the inside out. And that's an opportunity for you to do that and guess what type of fruits are actually shown in those images.
Yeah, and it leverages the super high resolution that we're able to get with that 900 megahertz, 21 Tesla MRI. Would you just tell people, compared to kind of a normal MRI that they're familiar with, just kind of put that in reference for people?
Sure. So a clinical MRI system, which most people would have some familiarity with, is a 3 Tesla system. And that is a great field. It's high field for clinical work. It's got the frontline type of magnet that you would use if you had a bump on the head, or if you had maybe a sprained knee or something like that. That's effectively 7 times, our 21 Tesla magnet is 7 times higher than that. And we get at least 7 times the signal to noise, the sensitivity for just looking at water distributions, which is what you do clinically. And you get even higher sensitivity if you look at other parts of the periodic chart. So we get the opportunity with that magnetic field to look at things like sodium, carbon, oxygen, other nuclei that are very biomedically relevant and areas of very intense study for myself and colleagues at the MagLab in a very unique way, so that we can see what's happening dynamically as well as with regards to the distribution of those biomolecularly important elements around the brain and around other parts of the body.
Hey, Sam, we've got a question that came in, which is actually a question I was thinking about, so I'm glad I have an excuse to ask this. And the question is, we're using fruits today. Can we use vegetables?
Absolutely. Vegetables are going to have DNA as well. So you can very easily use any type of vegetable. I've used green beans to do this. I've used lettuce. Works pretty well to do it. All you need to do is just make sure you smash up that cell wall and are able to get at that DNA. So yes, absolutely. Play with different types of vegetables as well. Usually you want to take the skin off, so it's a little bit easier to get at the insides. But otherwise, feel free to experiment. That's one of the great things about being able to do something simple like this, even in the house. You get to play with different types of ingredients and different types of experimental variables.
So Sam, just for any of our participants that are watching and are kind of thinking about, whether a future STEM career might be right for them, maybe they want to be a researcher or an engineer. Could you talk a little bit about, you are actually affiliated faculty at the College of Engineering, right? So how does being engineering faculty, and using magnets all mash up, right? Could you kind of explain that to our hopefully fledgling scientists in the audience?
Sure, my pleasure. So I am a biomedical engineer, and what that means is effectively as an engineer, I get to build solutions that bring the basic sciences, physics, chemistry, and biology, and apply them to human health. And that's the area that excites me the most. It's the type of work that I do. Hopefully will have an impact very directly on patient care in the future. We use the high fields in order to give us information about how effective those different treatments and those different approaches are that we're taking. And that's a critical link, because if we can do that in a test tube and move it into animal models and then move it into humans, then we have a way of demonstrating the effectiveness over a wide range, and that then gets us into clinical use and into an actual therapy that can be benefit to the public health and the public good much more rapidly. So biomedical engineers do that in particular, but I actually started as an electrical engineer. And I do quite a bit of electrical engineering still, and that's building different devices, building different detectors in order to carry out this type of work at high fields and high frequencies. And that's also a very interesting area. And engineering is, in my mind, one of those great fields for those people who like to fix things, who like to find solutions, and who like to understand fundamentals, but then apply those fundamentals. And when I was looking at a career, this was back in high school, of things that I could really approach, I really liked physics, I really liked chemistry, but I wanted to apply them. So I looked for the toughest engineering fields that I could, and I kind of attacked those. And in my mind, those are along the lines of electrical and biomedical engineering.
I've got a question. So you mentioned that the typical MRI in a hospital is about 3 Tesla, and the one at the lab is 21 Tesla. But I remember hearing that the strength of the field is not as important as the frequency of the magnet or the field that is being used. What's the frequency of a hospital MRI, and what's the highest frequency that we have available?
Sure. So the frequency of a 3 Tesla magnet for protons, and that's when you're looking at the water distribution in the body, which is what most clinical MRI is based on, is 128 megahertz. The 21 Tesla magnet, the proton frequency is 900 megahertz. And then we actually have magnets at the MagLab that go up to 36 Tesla. And that is equivalent to about 1.5 gigahertz. So we go from basically radio frequencies up to very high radio frequencies, up and almost into microwave frequencies. So it's a wide range of frequencies that we're looking at, and the magnetic field is very important. But being able to couple into the magnetic field by being able to put RF energy into a system and then listen to extract the RF energy that comes back is the most critical part. And that's actually in the terms of frequency.
I just posted last call for questions into the chat, so we'll just give everybody a second or two to see if there's any final thoughts, questions, things you have to know from Dr. Sam Grant. This has been any questions about how to do the experiment, or, you know, if you change something, would it change a result? There are no bad questions.
I'm going to go ahead and show how we're doing. We've got lots of DNA that's come out in the strawberry solution. Real cloudy now, you can kind of see it there. And even in the banana, if we go to that, you can see it at the interface as well. All the little bubbles that you see there are actually bubbles that are on top of that DNA. So that's actually extracted really well. So let me see if I can take a paper clip real quick and pull that out. And that's while anybody thinks about last minute questions, that's a good place to end.
I just thought of a question. So talking about fruits and vegetables and the DNA that's within them, I started thinking about what else could you test? What other DNA could you extract? Is there a food that wouldn't have DNA?
Well, I'll tell you what, anything that grows, anything that's living, is going to have DNA associated with it. So spices, if they come from vegetables or come from something that's grown, they'll even have DNA. But a lot of those, the DNA in those will be degraded because they're often cooked, or they're dried out, or they are somehow affected so that the DNA is denatured, which means it falls apart. Salt, obviously, and other minerals are not going to have DNA. And then anything that is cooked as far as a product, so looking at your green beans, for example, after you boiled them or after you steamed them, is not going to work nearly as well as when they're fresh. So they'll have DNA, but it's going to be harder to extract.
Okay, so I can extract chocolate DNA. That's a thing that we could do.
It's possible, but I think it's going to be real hard. My suggestion is probably just go ahead and try.
We've got a question that just came in. Someone asked, can the magnets at the MagLab harm humans? So can the magnetic fields harm us?
Well, that's one of the great things about magnetic resonance imaging, and it's one of the reasons why it's used fairly extensively in hospitals, is because it's not detrimental to humans. So as long as you take safety precautions with regards to how you use different types of metals in magnetic fields, or how you interact and move in the magnetic field, they're very safe. So as long as that is taken into account, you're usually in pretty good shape.
Those are great questions. And this has been such a fantastic session. We appreciate everyone joining us. Stay tuned after the webinar ends for a survey on this session. So as soon as we end the webinar, it'll trigger a survey. But I just want to thank Sam Grant and, of course, our partnership with the College of Engineering for Engineering Week. And don't miss, the future open house session scheduled for the rest of this week and into next week. We look forward to seeing you at a future session, even tomorrow. More engineers being featured and more fun activities. So come back. Thank you everyone for joining us. Take care.
Thanks everybody. Have a great night.